Thank you. Our next speaker is Mike Boland, who's going to talk about trying to find CSF biomarkers for STXBP1.
Thanks to the Foundation for inviting me to speak about some of our work at END to develop molecular biomarkers for STXBP1 disorders. I knew it was going to be difficult to follow Julie's talk. I didn't know it was going to be this hard. I will say I presented some of this work at a meeting recently, and some members of the FDA were there. And I said, so what do you think about this biosensor work for tremor? And they said, it looks like an endpoint to me. So we're pretty happy and really confident that we can develop this as something to measure in future interventional trials. And this is also piggybacking on the work that we're doing in the lab to identify molecular biomarkers. And so I think we need to understand the difference between outcome measures, endpoints, and biomarkers. They all have a different use in interventional trials. Outcome measures are what we're going to hear a lot about later. These are Bailey, GMFM. These are assessments of how a patient feels or functions. Whereas endpoints are analyzed statistics that can use to determine whether a study is successful or not. For instance, a 10% movement or increase in the 6-minute walk. Biomarkers, on the other hand, are objective measures that can indicate normal physiology and response to treatment. And they come in a few different flavors, different types of biomarkers that Wendy alluded to earlier in her terrific opening. And these are diagnostic biomarkers, prognostic or predictive biomarkers, but I think what we really want to focus on for interventional trials is a pharmacodynamic biomarker, and this is definitely the highest priority for phase 1-2 trials. And this gives us an indication of therapeutic target engagement as well as efficacy. And so this is one thing that we're really focusing on in a more targeted manner than the multi-omic approach that Wendy so nicely presented earlier. So I'm going to spend the majority of the time talking about our work to develop STXBP1 as a biomarker in human CSF, and I'll spend a few minutes at the end talking about some of our unbiased proteomics work in human blood and plasma to set the stage for Fawn's talk after the coffee break. So let's start with the CSF. So after a couple of years of trying to enrich for extracellular vesicles in normal pediatric cerebrospinal fluid and CSF, after trying to develop targeted antibody enrichment or affinity enrichment protocols to detect STXBP1 in CSF, we were just not having a ton of luck. So we decided to turn to an unbiased approach. This is the SEER platform, SEER proteomic platform in collaboration with the CHOP Penn Proteomics Core. And what this is it's a library of more than 250 nanoparticles that each has a different property, a different protein affinity and what have you. So you just treat your biomaterial, whether it be plasma or CSF, with these nanoparticles, and you get this protein corona on the nanoparticle that you can then take to downstream analyses like LC mass spec. And so doing this with, I'm just going to show a little bit of our data. We've done more than 30 samples, more than 30 pediatric normal CSF samples, but this is just kind of an idea of what this SEER proteomics can do. It's actually really amazing. This is only 300 microliters of CSF, and we can detect thousands of proteins, upwards of 5,000 different proteins in CSF. And the quality metrics are really good. There's always one outlier sample no matter what you do. And of course, in this one, it's control 5. But the quality control for this is very nice. And, you know, one thing that we're interested in is STXBP1 itself as a marker of target engagement and therapeutic efficacy. But also we recognize that, like we heard about yesterday, there are a lot of expression differences of syntaxin in haploinsufficiency models of STXBP1. And so we're interested in the different syntaxin isoforms, particularly 1A and 1B. 1B is the primary isoform that STXBP1 interacts with for synaptic vesicle exocytosis, and STX1A is the primary isoform for dense core particle release, which is another function of STXBP1 that we often overlook. And this is the exocytosis of neuromodulators or neuropeptides that have a signaling function within the brain and in peripheral tissues, actually. And so what I show here on the left is that we can reliably detect STX1A and 1B and STXBP1 at quantifiable levels in CSF. And we also detect a number of different of these neuropeptides that we're definitely interested in following up as potential surrogate biomarkers. Interestingly enough, we have trouble detecting neurofilament light chain, which is one neuromodulator that people really like to focus on. And for some reason with this platform, we had one sample that detected neurofilament light chain, but we have plenty of others to think about. We wanted to also test what is the reliability and the dynamic range of STXBP1 detection. And this goes back to, can we reliably detect, do we have the dynamic range and to detect STXBP1 haploinsufficiency at reproducible levels? And so we looked at 2 different biobanks on the CHOP campus. And again, we could reproducibly detect STXBP1, but the variability between the 2 biobanks was significantly different, as indicated by the power analysis that you see at the bottom, where samples in biobank had a log 2 fold change of 2, and that's about 9 samples to detect haploinsufficiency, whereas in biobank 2, there was quite a bit more variability, and you would need greater than 80 samples to detect haploinsufficiency. So it goes without saying that sample processing and storage is absolutely critical for reproducible results when you're talking about any proteomics using biomaterials. The next thing we asked was, can we detect STXBP1 and related neuromodulators reliably and consistently over time? The CHOP biobank has a large number of samples from patients of SMA because they get a CSF draw every time they get their ASO. And so we took 3 different SMA patients and sampled their CSF at 1, 4, and 8 months on the cadence of their ASO injections. And as you can see here, we can very reliably detect STXBP1, syntaxin, and these neuromodulators over time in normal pediatric CSF. So a lot of this unbiased work actually taught us, STXBP1 is there. We can detect it, but it also showed us which peptides fly the best on the mass spec. And using that knowledge, Steve Master and Fawn, who you're going to hear from in a minute, have started to develop custom aptamers to some of these peptides and using them to capture STXBP1 in CSF and in plasma in more of a diagnostic assay that we can really increase resolution and sensitivity. I think it's worth mentioning, and Ben mentioned this paper yesterday, or I should say Dr. Prosser mentioned this paper yesterday, and it's a good example that we're on the right track. where the Biogen AAV STXBP1 program did an experiment where they injected a PHPEB mice, neonatal mice with via ICV, and I guess it was AAV9. Sorry, I thought it was PHPEB. But after 9 weeks, they harvested cortical tissue and CSF from the mice, and if any of you know that mice have like 10 microliters of CSF, so to do this experiment, they probably had to kill about 300 mice at least. They were able to show an increase of STXBP1 after treatment in the cortex as well as in the CSF using this AAV-mediated gene therapy. And then on the right, all they're showing is that they can detect STXBP1 and syntaxin in human CSF, which we can as well. So I'll turn just in the last few minutes to a little bit of our work in blood and plasma. And, why do we want to think about blood biomarkers for largely CNS disorder? And this was alluded to in Wendy's talk, and I really kind of brought it home when I attended a symposium held by Combined Brain, where they looked at essentially an unbiased proteomic approach in plasma from more than 25 different neurodevelopmental disorders, and every single one of those disorders had significant dysregulation of immune-related processes. And so I think that there's definitely something to be said for looking at immune-related processes in neurodevelopmental disorders in the periphery as potential surrogate biomarkers if the target of interest isn't available. For instance, like SYNGAP. We cannot detect SYNGAP protein in CSF or blood using affinity enrichment, nanoparticle enrichment, what have you. And so I think for something like SYNGAP, we will need a surrogate biomarker, potentially one of these neuromodulatory biomarker proteins. So this is kind of a breakdown of the number of samples that we initially looked at, and this was funded and sourced by the STXBP1 Foundation and Combined Brain, where they had SomaLogic look at kind of a broad range of different proteins, and these are the disorders that were included in this large study. We looked at STXBP1. This is work that Janine did, and while we see a trend for haploinsufficiency in STXBP1, we just didn't have the sample size, particularly in the controls, to be able to reliably say that. We can detect the syntaxins in plasma, and many of these secreted neuropeptides, but none of them rose to significance in this sample size. So, and this was a question that Jan asked earlier: will a potential blood-based biomarker actually reflect a delivery that's a CNS delivery? And we want to try to tackle this question using the mouse model and AAV gene replacement, where we will do ICV injection of haploinsufficient STXBP1 mice, and then harvest tissue and blood, and then perform both targeted and unbiased proteomics to see if there are some sort of treatment responsive biomarkers in the blood that we might be able to hang our hat on. So Wendy also mentioned this, and it's really important idea that do levels of STXBP1 or surrogate biomarkers actually correlate with clinical severity? Can we identify biomarkers that are cross-species compatible, which would be amazing for therapeutic testing, given the strength of the STXBP1 mouse model phenotypes? And obviously, is STXBP1 haploinsufficiency reflected in CSF? That's the hardest question to answer, because it's very difficult to get CSF from STXBP1 patients. So we're gonna work on that, but that one's gonna be tough. I will end by thanking Lucas, my son, as well as a number of people that have contributed a lot to this work and the entire N team at Pen and Shop. Oops. I'll take questions.
Hey, that was awesome. So, as I was following your talk, you made the comment about how the biobank matters a lot for the reproducibility, and then you showed the really nice data showing that within the same biobank, you actually get good longitudinal data. I guess like balancing those two things, like how far away do you think we actually are from just like being confident that you can measure a, let's say a 50% increase in the same biobank longitudinally? For instance, like is the assay actually already there if you're at the same biobank, or do you have to improve?
I think that with the unbiased approach, it might be a little more difficult, but with a targeted approach using custom aptamers, I think you'll have much, much greater sensitivity and detection power, and that's something that Fawn is going to talk about in the next talk. Yeah. I think a more a lot of this unbiased stuff was just to make us feel better, because we spent so long trying to adopt an affinity enrichment or extracellular vesicle enrichment and all of this, and then actually it was a meeting with Fawn and Steve Master at the Center for Diagnostic Innovation where we said, look, we can detect STXBP1 in all of these different peptides and its binding partner with somomers. And he said, we'll just start using somomers. And they made a custom somomer to a peptide that was most frequently observed in all of these unbiased samples on the mass spec. So I think we're getting closer to answering that question.
Okay. I guess I was also just maybe surprised but impressed in a sense that the longitudinal data seemed so good. And just the intuition actually is that, you know, CSF stuff depends a lot on like circadian rhythm even. Like things can change a lot even on a moment-to-moment basis.
Yeah.
And so, yeah, it was just...
I was surprised too.
Yeah, okay.
Yeah. Rude, thank God.
Yeah, of course.
Okay.
Thank you so much. Great presentation. I actually have a super naive question. What is your idea about the detection of STXBP1 and syntexin in the CSF? So other than cell death, so are they secreted proteins in your opinion? Or?
No, I don't think they're secreted proteins. I think they're probably leaked into the CSF through extracellular vesicles, and I think that's probably where they are in the plasma too.
Right. And did I get you right? Is that isolating EVs is really tough from
Yeah, it's tough. We had to use like 10 mils of CSF to see a signal. Now we can do 300 microliters.
And in this way, did you detect any potential EV proteins, so the CD63s or ALEX, or any protein that you can that will give you confidence that the STXBP1 that you measure is actually coming from exosomes?
We haven't looked into that really closely yet, but I think Fawn will probably allude to a little of that.
Great, thanks so much.
Yeah, thanks, Ruud.
Yeah, I have a question. Do you think CSF is more relevant with a ependymal layer or less relevant with
Yeah, absolutely. I think the ependymal layer and what goes on there is going to play a large role in
how relevant
in the
with
CSF content.
The disease, like because the disease is more relevant with the cortical structure.
Yeah, I don't know. Yeah, I just don't know the answer to that question. If the ependymal layer plays more of a role in the CSF content than cortex, I just really don't know. Thanks.